PhD is in the life sciences. I work at the Discovery Institute Center for Science and Culture here in Seattle, Washington, which is a nonprofit organization that funds research into and promotes the public understanding of intelligent design, which is the study of patterns in nature that bear hallmarks of design. In addition to my interest in intelligent design, I also have a strong interest in New Testament scholarship, the case for the resurrection, other arguments that bear on the truth of Christianity in particular. You can find a personal website at jonathanmclatchie.com, which houses many of my videos and articles relating to intelligent design as well as biblical scholarship and other issues. And you can also find the Discovery Institute's website at discovery.org. And if you want to look at for the Center for Science and Culture, which is the intelligent design program of the Discovery Institute, you can go to discovery.org/ID. And you can also send me a private email at jmclatchie@discovery.org. So as I mentioned, my interests are very broad. I'm interested in the sciences as well as in biblical scholarship. And in 2021, I launched this organization called talkaboutdoubts.com, which is an online ministry that specializes in mentoring Christians who struggle with doubts about the truth claims of Christianity, as well as ex-Christians who want to explore whether there's a rational path back to faith. And so if anyone fits into those categories and would like to reach out, basically the way it works is someone comes to our website, they submit a contact form, and then we distribute that to one of our volunteer experts who will then get in touch privately to set up an online Zoom meeting to talk about their doubts with them in confidence. And so we have people with expertise in biblical archaeology, textual criticism, New Testament, Old Testament scholarship, philosophy, biblical archaeology, physics, biology, we have pastors, psychologists, etc. And so our goal is to take people who are struggling with doubts or have sincere and honest questions about the truth of Christianity and connect them with some of the world's leading and finest experts on those topics. So again, talkaboutdoubts.com. So let me just lay out some agenda for the course of this evening's discussion. We'll talk about, first, we'll lay out what is intelligent design for those of you who maybe haven't come across the concept before. How many of you are familiar to some extent with intelligent design already? Yeah, great majority of you, excellent. Okay, we'll talk about nature's suitability for advanced life. We'll talk about the irreducible complexity on a cellular level. We'll talk about the waiting time problem, and then finally we'll draw our conclusions. And at any point, you're welcome to interrupt me or raise your hand, and I can field questions as we go. Otherwise, we will also field questions at the end as well. And at the end of each section, I will also have some discussion questions for you to discuss with people around you as well. Okay, so I want to keep this fairly interactive. So then let's begin with a discussion of what is intelligent design. So how do we detect design in our everyday experience? Well, here we see a cliff face. How many of you think that this cliff face is a product of design, conscious intent? How many of you think that there was a chiseler who took it upon himself to chisel this particular rock formation into this cliff? Yeah, one person, any other takers? Okay. No, we don't infer that this is the product of design. We infer this is very probably the product of wind or erosion over long periods of time. Okay, so what about this one though? This is Mount Rushmore in South Dakota, and we of course recognize the faces of 4 presidents of the United States that are chiseled onto this cliff face. And of course this very obviously is the product of conscious design. Well, what distinguishes this formation from this previous one that we looked at? Obviously, they're both very complex conformations. Why do we recognize design in this cliff face but not the other? Well, you'll notice that it's not merely the improbability, but it is also that the second cliff face has conformity to an independently given pattern. It has what we call specification. Okay, so how do we talk about how should we think about evidence, the nature of evidence? When I say that such and such is evidence for intelligent design, what really am I saying? Well, to help illustrate this, let's think about a court scene where you have an accused, someone who's charged with some homicide, for example. And let's say that the forensic scientist or the detective comes forward and presents the murder weapon that was used to perpetrate the crime. And the detective notes that on the handle of the murder weapon are the accused fingerprints. Now, does that prove that the defendant is guilty? No. not necessarily. There could potentially be other explanations for how his fingerprints came to be on the murder weapon. But it is evidence that he is guilty. Why is that the case? Well, the presence of the accused's fingerprints on the murder weapon is evidence for the guilt hypothesis because it is more probable that his fingerprints would be on the murder weapon, given that he's guilty, than given that he's not guilty. Right? So you might imagine that there could be other evidence that is brought to bear in this crime scene. Perhaps there are tread marks close to where the homicide was committed, which match the shoes of the accused. Or perhaps there are fabrics that are discovered nearby which match a sweatshirt that the accused is known to possess, and so forth. Now, you might, the defense attorney might say, well, how many other people do you think have that particular make of shoes? How many other people do you think have that particular sweatshirt in the city? And that would be true, but each of these is more probable, given that he is in fact the one we're looking for, than otherwise. And so the defense essentially has to scotch tape together a lot of different auxiliary hypotheses to make the data fit on the hypothesis that he's not guilty, whereas you could just explain all the evidence in one fell swoop by postulating, well, maybe the defendant's actually guilty. And that's what we're doing this evening. This is a cumulative case from multiple lines of evidence that all convergently point in the direction of design. And so the naturalist, the one who denies design and argues that this is merely the appearance of design, has to postulate lots of different auxiliary hypotheses to make all the data fit on naturalism, whereas design takes care of all of these pieces of evidence in one fell swoop. And so it's, I would contend, the better explanation. So going back to our Mount Rushmore example, why is this evidence of design rather than the processes of erosion and other natural phenomena? Well, as I said, these faces conform to an independently given pattern, namely that we recognize these faces as corresponding to 4 of the presidents of this country. And so that is more probable, or less surprising, to put it another way, on the hypothesis of design than it would be on the falsity of that hypothesis. Okay, so let's take another example. How many of you think that this beach is the product of design? How many of you think that the grains of sand on this beach are very carefully arranged by an intelligent agent? Well, it's not very likely, right? What about this one, though, where you see Mary loves John on the sand? Well, that's obviously the product of design because it has specificity. Or what about this one? Again, it has specificity. It's not merely that it's a complex arrangement, but it conforms to some independently given pattern. Now, there's a very common misconception about the nature of theory and prediction, or theory and evidence. And that is, the misconception is that in order for a piece of data to count as evidence for a hypothesis, the hypothesis has to strongly predict the data. This is false. To illustrate this, let me give an analogy. Imagine that you're hiking, and you're far away from roads and civilizations, in the middle of a forest someplace, and you come across this cabin. And at first it looks like it's not inhabited, it looks like it's run down, there's no one living there, but you decide to inspect nonetheless. And as you prise open the door, you find this cup of Earl Grey tea that is still steeping. It is not at room temperature. Now, on the hypothesis that the cabin's inhabited, would you strongly predict that you'd find specifically Earl Grey tea inside this cabin? No, right? Could be coffee, could be other types of tea, English breakfast tea, or chai tea, or there might be no tea. So the hypothesis that the cabin's inhabited does not strongly predict this data. But nonetheless, the hypothesis the cabin's inhabited makes this far more probable than it would have otherwise been if the cabin's not inhabited. Does that make sense? Yes?
Yeah, I just want to make sure I understand a couple of things as you're giving us your lecture. First, what you're trying to show us is that through proving that intelligent design exists, that proves the existence of God, and if that is what you are trying to show us, is it a particular version of God? I think it's pretty clear it's a Christian God, but is it like the Protestant version of God,
Okay.
the Catholic version of God,
Right.
the Orthodox, or in other Christian sake? I'm just trying to understand where you're coming from with this.
Sure, absolutely. So the evidence that I'm going to be adducing in this presentation, I think, is very powerful evidence for intelligent design, which is in turn evidence for theism, the existence of God, because the evidence of design in nature is far more probable or far less surprising if God exists than if he does not exist. Now you ask the question about, well, which God are we talking about? Are we talking about the God of Judeo-Christianity, or are we talking about the Islamic God, or what are we?
Given the name of the organization putting this on, I'm sure that you're Christian and you're trying to show that a Christian God exists, and please don't take this as some sort of subtle put down or something. I'm just respectfully asking you this. I'm just wondering, because versions of Christianity differ greatly, and so, I'm just trying to understand which version of Christianity, intelligent design proves.
No, so first of all, I don't like use of the word prove in this context, because you only really get proof in pure mathematics, right? 2 plus 2 plus 4 is a proof. But we're talking about evidence. So in science, nothing is proven, right, in the rigorous deductive mathematical sense, right? But I would argue that given the data, we can be immensely confident in the reality of design. Now, coming to your other question about Christianity versus other religions and various sects within Christianity and so forth. So I'm a Christian. I think that there is an avalanche of public evidence that substantiates and justifies the truth of Christianity, but that is independent. Okay, let me finish. But that is independent of the case for intelligent design. So the case for intelligent design, I think, provides powerful reasons to think that God exists. It contributes to a broader cumulative case for theism. There's also the cosmic fine-tuning arguments, the Kalam cosmological arguments, and so forth. So it contributes to a broader cumulative case for theism. But intelligent design, in its purest sense, doesn't really distinguish between the major world religions, right? Now, I do think that you can justify strongly the truth claims of Christianity. If you would like to have that discussion, we can have it later. But that would be a set of arguments that would be independent of what I'm presenting this evening.
Okay. I just didn't quite hear that word all the way back there, defense. So I was just asking you what word you used, that's all.
Okay. Cool. Anyone else?
Well, could I?
Yes.
Because there was sort of an assumption built into your question. You said, given the name of the organization, you use the word, you assume that he's trying to prove the Christian God. And obviously, I assume the organization is Discovery Institute, is that right?
Oh, no, I meant that it's...
Yeah, he meant Ratio Christi.
Oh, okay, got you, okay, I'm sorry, okay.
Yeah.
Okay, got you, okay, I'm sorry.
Yeah.
Yeah, I would say Ratio Christi is bringing in different types of speakers, showing different points of view, not necessarily proving Christianity outright, but we like to hear different things and discuss them. Yeah.
Okay, but it's pretty clear your organization is associated with Christianity.
Oh, Ratio Christi definitely.
So Ratio Christi itself is a Protestant Christian organization. However, we want to have lots of different viewpoints.
Right.
Yeah, that's all I was doing, is saying, because a Christian organization's putting it on, I figured, you're talking about a Christian God.
Right, but all I'm saying is that the arguments I'm presenting tonight don't get you that far, and they're not intended to. Okay. Okay. Any other comments or questions before we go? Okay. Right. So let me just wrap up this section then by recommending these 3 books which develop this argument in more detail. This recently published second edition of The Design Inference by William Dembski and his co-author Winston Newart, which basically develops the argument from specified complexity. The new edition also discusses how this could be hashed out in Bayesian terms, like I've done. The second book there is No Free Lunch, which is a sequel to the first edition of The Design Inference, Why Specified Complexity Cannot Be Produced Without Intelligence is the subtitle. And then the third book on the screen there is Signature in the Cell, DNA and the Evidence for Intelligent Design by Stephen Meyer, which focuses on the origins of life and how that points to design, and in particular developing rigorous methodology based on inference to the best explanation for inferring design in the natural world. Okay, so let's do some discussion questions. Maybe you could break into groups of 3 or 4 or something like that to discuss these. So based on the inference, based on the methods of inference described in this section, how would you make the argument to a friend that the information content found in DNA is evidence of design? And does this argument commit a God of the gaps fallacy? Why or why not? So maybe break into groups of 3 or 4, and I'll give you maybe 5 or so minutes to discuss.
I just came here to hear him talk.
Well, I think the idea would be that the information comes from the web. You see information being created, it always traces back to intelligent agents. That's what I would see.
We can't come with like an explanation for why a super complex beach or a super complex rock formation exists. Therefore, so like it probably can be. I think with DNA, I'm not like an expert on DNA, but I know DNA is pretty good. So it's like yeah.
So much so that like that's what the
But if we just go over and say, hey, DNA is super complicated, you create and clearly that shows intelligent design because DNA is complicated, therefore God, it's like, it needs a bit of a zells here, but.
There would definitely be a loop that. I think the most you could get from the information would be if there was a mind there. You know, if you were to go further and say, well, this is the God of the Bible, and this is Allah, or whoever, that would be
Okay, guys, just need your
And what do you think is it goes gaps?
just saw the
Based on
notices.
the, oh, wait, what's the first one, the second? Because I'm thinking it's like, DNA evidence design, do you think like attributing that to God is like God of the gaps?
It gets asked the argument that's constructed so far. The argument that he's put forward so far. The complexity conforming to a specific pattern. Is that just fancy language covering other gaps?
do that again in different ways. It doesn't go beyond what
Somebody looking for internet signals.
Okay, so let's look at these questions then. So on the first question, so based on the methods of inference described in this section, how would you make the argument to a friend that the information content found in DNA is evidence of design? Any thoughts? Yeah.
You would have to construct some sort of argument that would be saying that there's no, to prove that correlations between like something happening exterior, non-living process or non-thinking process, to the changing of nucleotide sequences, that cannot produce the kind of complexity that you see.
Okay. Anything else? So basically the approach that I would take here is to argue that information content in every realm of experience is habitually associated with conscious agency, right? Conscious design. And so when we find information content intrinsic to the hereditary molecules of DNA and RNA, the best or most reasonable explanation is that it too arose by virtue of an intelligent cause. Or put another way, the information content of the cell is not hugely surprising if we suppose a mind is involved. but is wildly surprising otherwise. And so you have this overwhelmingly top-heavy likelihood ratio that confirms design. Does that make sense? Any questions on that? Okay, what about this second question? Does this argument commit a God of the gaps fallacy? Why or why not? Who thinks it does commit a God of the gaps fallacy? Okay, so those that said yes, which is I think like 3 of you, why do you think that? Yeah.
Well, I think it definitely can be. Like, you can't just go over and say DNA is super complicated and there's like, if you stretch it all out and go all the way to Pluto, something like that, I think it's the same with DNA, right? So it's like we can't go over and say, oh, DNA is super complicated, it's super complex, and we can't explain why it's so complex, therefore God, because that's like I don't even know, like ancient superstition of like, oh, we can't explain why lightning strikes, therefore there must be like a God behind it. So you need to like be very like precise and careful with like the argument that you make. So you can't just go over and say, well, just look at the world and see how complicated it is. Clearly there's a God. It's like, you can't do that, but maybe you can go over and like appeal to like the natural world and like talk about it more. But simply pointing to DNA, and it's like, oh, DNA is super complicated. God made it. It's like the product of God. It's like,
Okay.
I think it's God of the gaps.
Anyone else? Any of you did?
I was kind of thinking on the same lines. I was trying to pick apart earlier, like what is the God of the gaps fallacy?
Okay.
But I guess, yeah, kind of like both. If I'm just thinking super complicated.
Did anyone have something to add? Yep.
I'm not sure if it is or not, but assuming it is, I don't necessarily think that it's not so strong of a God of the gaps fallacy as like we don't understand how lightning works, because it is talking about information and some form of ordered knowledge. But even if that is a God of the gaps argument, that doesn't necessarily make it invalid, it just makes it less strong. So coupling it with other theistic arguments is just building a case.
Okay. Anyone else? Okay, so the God of the gaps argument essentially says we can't explain feature X, so we'll just plug in God as a placeholder for that of which we are ignorant, and then as science advances, the God of the gaps argument has to retreat with each new advance in scientific discovery, right? But as we've seen already, intelligent design doesn't work that way. Whatever else might be wrong with the design inference, it's not wrong because it's God of the gaps argument, because it's actually making a positive case. It's saying these features are less surprising or more probable, more expected, on the hypothesis of design than on the falsity of the design hypothesis. And so it tends to raise the probability of design relative to what it would have otherwise been, right? And that is the nature of evidence. Evidence, by its definition, changes the probability of a proposition being true, right? And so if a piece of evidence a piece of data raises the probability of design, then by definition it is evidence of design, right? Yes.
So why couldn't that exact argument have just been made about about lightning, X number of years ago when we didn't know what produced it?
Yeah, so you'd have to make the argument that lightning is less surprising on theism. And I've never seen that really rigorously developed. So there are all sorts of things that we still don't understand to this day, and yet we don't. They don't justify an inference to design. So, for example, in physics, the wave-particle duality of photons, we don't understand that, right? We don't understand what it is, what it means to say that photons are both a wave and a particle. But we know it's true. But there's no reason to think that that is less surprising on theism, right? It seems that for all we know, it's equally probable on theism or atheism. Yeah.
So it's contextually specific to intelligent design because of the information content that we see.
Yeah, things that we associate with conscious activity, such as information and other features, as we'll see.
Because we don't necessarily attribute lightning strikes to consciousness.
Exactly. Okay. Yes.
What does less surprising on theism mean?
Meaning that, or put it another way, it's more probable or better expected on theism. Okay? All right. So let's go to the second section then. So nature suitability for advanced life. This is a species of fine-tuning argument. So let me just summarize the take-home from this section before I go and fill in some details. So there are, as I'm going to show, there are far more numerous ways in which the universe might have been that are non-conducive to life than those that are life-friendly. Right? There's a lot more ways that the universe would have been prohibitive to life than those which are actually conducive to it. On theism, it's not particularly surprising that the universe would be hospitable to embodied conscious beings like ourselves. Because if God exists, then it's not especially improbable that you might create a community of embodied agents that can engage in moral decision making, molding and shaping their character, growing in morally significant ways, and so forth. And is being embodied specifically that really accentuates that ability to engage in moral decision making. On atheism, on the other hand, it is wildly surprising that the universe would be supportive of such beings. And therefore, the following data tends to confirm theism. So there's a lot of data I could go through. I could literally be here till Christmas giving you examples of evidence for theism from this category. But I'm just going to give you a couple of examples to whet your appetite, and I'll give you resource recommendations where you can get more. So, of course, many of you, I'm sure, will recognize this chart. It's a periodic table of elements, if you've taken a chemistry class. And it's specifically the non-metal atoms are highlighted in purple there on the screen. Namely, in particular, carbon, hydrogen, oxygen, nitrogen, that make up the material substances of the cell, right? And these are the only atoms that you could use to build a biochemical system. And there's a good reason for this, namely, that they make strong, stable, directional chemical bonds. And critically, these covalent bonds give molecules with shape. And it is shape that is the essence of biochemistry. If you want to make, if you want an enzyme to plug into its substrate, it has to have a particular shape, right? If you want a protein to be part of a larger molecular machine, it has to be very specifically crafted. It has to have a very particular shape. So what's fascinating is that these atoms have an electronegativity such that the attraction of electrons for hydrogen and carbon is very similar. So this means that you generate, by putting carbon and hydrogen together, non-polar molecules. So what does that mean? So carbon and hydrogen have a similar value for electronegativity, which is essentially how strongly atoms pull electrons towards the nucleus. And when you form a covalent bond, the pairs of electrons get shared between the atoms. So this means that they are able to share electrons, and that creates a covalent bond. And since carbon and hydrogen have very similar electronegativity, the electrons are shared equally, right? And the bond is called non-polar, meaning that they're shared equally between the two atoms, and they form a covalent bond. But if you put oxygen and hydrogen together, you get what we call a polar molecule, where the electrons are not shared equally. And this is critical to the whole organization of the cell because it gives you what we call the hydrophobic force. And it's a hydrophobic force that organizes the higher levels, the higher structure of the biological realm. So you see in this figure here, this is a protein. Proteins, as you probably know, or you may know, are chains of amino acids which are held together by peptide bonds. And the protein will collapse into a three-dimensional structure as determined by the properties of the side chains of the amino acid subunits, right? And there are polar side chains and there are non-polar side chains. These are also known as hydrophilic and hydrophobic side chains. The hydrophobic side chains, they don't like to interact with water, hence hydrophobic, and so they actually get buried inside the interior of the molecule. Whereas the hydrophilic side chains, they face the exterior of the protein, and they form hydrogen bonds with water. Okay, it's also critical for the formation of the phospholipid bilayer that forms the cell membrane. So they have a hydrophilic head and a hydrophobic tail. So this is critical for the formation of protein structures as well as the cell membrane. Now, so Charles Tanford, who was a biochemist, well-known biochemist, he says, describing the discovery of how proteins fold, he writes, and I quote, "The hydrophobic force is the energetically dominant force for containment, adhesion, etc. in all life processes. This means that the entire nature of life as we know it is a slave to the hydrogen-bonded structure of liquid water." Now, why is this evidence of design? Well, it's a remarkably fortuitous coincidence that the very atoms that give you stable, defined shapes from which you can build macromolecules also give you the hydrophobic force, which is the key to assembling them into higher three-dimensional forms. Nature does not owe us that coincidence, and yet it's there. And if it were not for that coincidence, life could not exist. Let's take another example. The sun, as you know, emits light, and the oxygen that we breathe is generated by a process called photosynthesis. in the chloroplasts of green plants, a process that's energized by this light from the sun. And remarkably, the radiation emitted by the sun exhibits several remarkable coincidences that make life possible. There are many forms of radiation that make up the electromagnetic spectrum, and each possesses a different wavelength. So here's a diagram illustrating the electromagnetic spectrum, and you can see that in this very large range of different radiation wavelengths, you have this very infinitesimally narrow band that we call visual light. This band is so narrow that it has been compared to perhaps a few playing cards in a stretch of cards, or a stack of cards stretching all the way to the Andromeda Galaxy. So this is a very, very narrow band. So it's a remarkably fortuitous coincidence that nearly half of the sun's radiant output happens to fall in that very, very narrow range. And if it were not for that coincidence, photosynthesis and photochemistry would not be possible, which would make all forms of aerobic life impossible. Yes?
Is that a statement about our particular star or about stars generally?
About stars generally, including the sun. Yeah.
If that light band was so prevalent, wouldn't it make sense for things to evolve taking advantage of that light band rather than the other one?
Well, the problem is that you're not going to get aerobic forms of life, which is, as far as I can tell, it is essential, critical for complex advanced life forms. So you might get anaerobic life forms that stay like single-celled and don't have mitochondria, chloroplasts, and so forth. And so you're not going to get multicellularity, because it was really the mitochondria that drove the transition from prokaryotes to eukaryotes. So you're going to stay very simple without being able to perform photosynthesis on Earth. Yes?
And the statement you just made about the contingency upon this visible spectrum being prevalent is due to photosynthesis. Are you claiming that there are no possible protein forms which can harness either larger or shorter wavelength radiation to do something similar to
I think it's very improbable. I mean, the other forms of radiation, like X-rays and gamma rays, are very detrimental to life, which is why the atmosphere absorbs them. Cyanobacteria are a form of bacteria that actually perform photosynthesis, and they did photosynthesis long before chloroplasts were on the scene, long before green plants were on the scene. And it's thought that cyanobacteria oxygenated the Earth's atmosphere, which allowed for the production of aerobic life forms that can perform cellular respiration and so forth. But without being able to perform photochemistry, it's game over for advanced life, certainly. There's also an adjacent near-infrared band, which is basically makes up the other half, or most of the other half, of the sun's radiant output. So this other half, the sun's radiant output, lies primarily in this other infinitesimally small region of the spectrum that's adjacent to the visual region. And this infrared radiation provides approximately half of the essential heat that is needed to warm the atmosphere of our planet. Michael Denton, who's a medical geneticist, in his book The Miracle of Man, says that without it, Earth's entire surface would be a frozen wilderness, far colder than the Antarctic. Of course, it's not just enough to get visual light, it also has to be able to penetrate the Earth's atmosphere. And so, again, the Earth's atmosphere is composed such that most of the visible window gets through, most of the visual light from the sun gets through to the Earth's surface. And it also absorbs enough of the infrared radiation to warm our planet to the conditions needed for photosynthesis to take place. It also absorbs the harmful radiation like X-rays and gamma rays and so forth. The light also has to be able to penetrate the water, not only because some plants are underneath the sea, but also because the light has to be able to traverse through the aqueous cytoplasm of the cell in order to arrive at the chloroplasts. and therefore be involved in photosynthesis. And so, again, it's a remarkably fortuitous coincidence that water is translucent to visual light. So again, let me summarize the take-home from this section. There are, this is just the peak of the iceberg, but hopefully this begins to show you that there are far more numerous ways in which the universe might have been that are not conducive to life than those which are life-friendly. On theism, it's not particularly surprising the universe would be hospitable to embodied conscious beings like ourselves. On atheism, though, it's wildly surprising, and so the foregoing data tends to confirm theism. If you would like to read more on these arguments and many others, I recommend these books, The Miracle of Men, The Miracle of the Cell, Children of Light, and The Wonder of Water, all by Michael Denton. My favorite of those is The Miracle of the Cell, which talks about the fine-tuning of the periodic table of elements, which is incredibly fascinating. So let's turn then to a couple of discussion questions. How would you evaluate the strength of the argument in this section? And how might someone's prior philosophical assumptions affect their evaluation of this argument and its implications? So let's turn to the first one. So how would you evaluate the strength of the argument that I've put forward here? Any thoughts? Yeah.
Yeah, I guess for me, maybe where you can kind of explain a little bit more, what's tripping me up is like, when I think about life existing, obviously there's a lot of elements that you've showed us, that water and light and the atmosphere, that all had to come together perfectly to allow life to exist. And the argument here is that they didn't just come together, by chance, but that there's a greater being who's helping all of those things fit so perfectly together. But then when I think about like the universe and space and how there's billions of planets and it goes on forever, like eventually, statistically, if planets in space go on infinitely, and there's a super, 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 super small probability that all of those elements are going to overlap, eventually they will, and here they did, and now here we are. So I guess that's kind of the way that maybe I'm seeing it sometimes, so I'm just curious what you're...
So a couple of points here. First of all, I think that the number of conditions needed for a life-friendly planet are so massive that I think that it even swamps the probabilistic resources afforded by our entire universe, even though there's many hundreds of billions of stars and so forth, planets. I also that argument doesn't even pertain to say the fine-tuning of the periodic table of elements, for example, like the non-metal atoms, carbon, hydrogen, oxygen, nitrogen, and their properties and so forth. And that was just the peak of the iceberg. There's a lot more there. These are universal properties. Furthermore, it's not just the sun that emits radiation that falls within that very narrow band, but actually most stars throughout the universe do as well. So I don't find that rebuttal to be particularly compelling. Yes?
I think that the one place where I see kind of an alternative explanation would, essentially, there's a couple things, obviously not like the fine-tuning of positioning of the Earth or the fundamental way that chemistry works, but things like the light gap and the part about photosynthesis. Could some sorts of evolutionary naturalistic hypotheses account for that, such as like the Gaia hypothesis, talking about like what gaps are available? Like that seems like it could be a function of the atmospheric composition, which could be a product of the biosphere itself, or is that just like naturalizing intelligence?
Well, that would be a form of intelligent design, if I'm understanding you correctly.
Okay, but it would just be the biosphere is what would make the intelligent thing.
Yeah, and then of course, since the universe is contingent and hasn't always been here, that would beg the question of because it's very difficult, I think, on naturalism to account for the origins of intelligence or teleology out of matter. This is why I think consciousness, by the way, is one of the most powerful arguments for theism. And if our universe itself is conscious, which I think you're proposing, and the universe is contingent and hasn't always been here, there's overwhelming evidence, I think, the universe hasn't always been here, then it begs the question of where it arose from. No, not really at all. I'd be really fascinated. I mean, is it based on this on DNA? Is it got the same? If it's got the same genetic code as us, then that would be even more evidence of design, for example, right? So it would depend on the particulars there, what I'd make of that. Okay. Oops. Okay, so let's move on then. So irreducible complexity on cellular level. How many of you have heard of irreducible complexity before? Yeah, a few of you. So irreducible complexity was actually a test that was put forward by Charles Darwin in The Origin of Species, published in 1859. And he says, and I quote, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down." Right? And so, in other words, if there were such a system where you required multiple different subfunctions to work together in unison to bring about some higher level objective, then it strains credulity that you could arrive at that by unguided process of successive modifications over long periods of time to arrive at that complex endpoint. So Michael Behe, who's a biochemist from Lehigh University, in 1996 he published a book called Darwin's Black Box, where he sought to take up Darwin's challenge. And he says, by irreducible complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, where the removal of any one of the parts causes the system to effectively cease functioning, right? So to help us understand this, he put forward an example from everyday use, namely the mousetrap. And the mousetrap has a number of different parts. You have a platform, you have a spring, you have a hammer, you have a hold-down bar, you have a catch, you have stables that hold the trap together. And if any of these parts is missing, you don't have a trap that works half as well as it used to, or a quarter as well as it used to, but it's broken, right? And in fact, the trap might still not work if the holding bar is too short or the spring too lightweight. So the parts even themselves have to be very carefully crafted to fit together to form this contraption, namely the mousetrap. Are there examples? Why is this evidence of design? Well, explaining the origins of irreducibly complex features requires a cause that possesses foresight. Why? Well, only intelligence can visualize a complex higher level objective and bring together everything that's needed to actualize that end goal. Unguided stochastic processes can't. With natural selection is not only a blind watchmaker, it's an unconscious watchmaker. So William Paley, who's an intellectual hero of mine, in his most famous book, Natural Theology, he says, and I quote, "In crossing a heath, suppose I pitched my foot against a stone and were asked how the stone came to be there. I might possibly answer that for anything I knew to the contrary, it had lain there forever. Nor would it perhaps be very easy to show the absurdity of this answer. But suppose I had found a watch upon the ground, and it should be inquired how the watch happened to be in that place. I should hardly think of the answer I had before given, that for anything I knew the watch might have always been there. There must have existed at some time, at some place or other, an artificer or artificers who formed the watch for the purpose which you find it actually to answer, who comprehended its construction and designed its use," right? And so this is making an inference to design. You're walking along a beach, you find a stone, you don't really make much of it, but if you find a watch that has many interacting parts that contribute to a higher level function, we immediately infer design as the best explanation. Now, yeah?
Is that just because we find the watch more useful than a rock?
Well, a rock can be very useful if you want to kill someone, you throw it and hit someone in the head and take them out. So, I mean, it depends. The prehistoric people found great utility in stones. That's what we mean by the Stone Age. So, it's not necessarily utility, but the watch has many different interacting parts that have to be very specifically crafted to fulfill a higher level function. Yes?
How does the interacting parts of a watch intrinsically differ from the interacting chemicals in the lattice structure of the rock and the electrons and protons and everything?
I'm sorry, say that again?
So we think of a watch's design because it's macroscopic parts that are fit together for a specific purpose. But in some sense, a rock is the same thing, just on a subatomic level, or at least a molecular level.
But I think the difference is, and I'll let Casey speak to this with his geology background. So a stone is uniform, right? There's regularity, repeating patterns. Whereas with a watch, you find complex and specified irregularity, and that's also what I would never find in life. Casey, am I correct there? Anything to add there?
I would agree with what you said. I mean, rocks are made of crystals, minerals, and minerals, they are specified. You know, there's a repeating pattern in a crystal structure, but they're not complex, because basically if you have basically a pool of magma of a particular chemical composition and you cool it below its freezing temperature, you're guaranteed, according to the laws of chemistry and the atomic packing and bonding, you're going to form those crystal structures. It's a natural phenomenon, and so it's not really complexity is related to unlikelihood. If something is highly likely, it's not very complex. So this would not be the kind of specified complexity. But like Jonathan said, with the watch, you get interacting parts and movement and specified complexity.
Right, exactly. Okay, so are there examples of this sort of system in life? Well, you bet. Let's take an example. So this is one of my favorite examples of irreducible complexity. This is the DNA replication machinery. I'm going to show a short 1-minute animation by an Australian animator called Drew Berry, which illustrates this incredible process. This is the process by which our DNA, our genetic instructions in our cells, are copied in preparation for cell division.
DNA replication is carried out by a molecular machine called the replisome that pulls apart the double helix and makes an exact copy of each strand. In this visualization, we're going to examine the mechanism for copying DNA. The enzyme DNA polymerase synthesizes DNA molecules, using the original molecule as a template for the production of a complementary strand. The genetic code in the template strands is paired to free-floating bases, matching cytosine to guanine and adenine to thymine. DNA polymerase creates an exact copy of your DNA code, making less than one mistake in a billion bases.
So as you saw in the animation, a DNA replication is made up of a lot of different proteins that have to be very specifically and carefully crafted to work together to bring about a higher level objective, namely replicating the DNA to prepare for cell division. It occurs in a semi-conservative way. That means that each parent strand is going to serve as a template for the synthesis of a new donor molecule. Okay? DNA synthesis is carried out by a group of proteins that act together as a replication machine. And if any of these parts is missing, you don't have a replication machinery that works half as efficiently as it used to, or a quarter as efficiently as it used to, but it's completely broken. So let's look at a few examples of what would happen if we were to miss or lose any of these proteins. So what about topoisomerase? How many know what topoisomerase does? What does topoisomerase do?
It, if I'm remembering it correctly, it changes the topology of the like the twisting of the DNA.
Yeah, so as DNA is being replicated, sometimes because of the torsional stress, it can induce supercoils. Think about an old telephone cord that forms a supercoil, and that, if left unchecked, will result in the DNA eventually breaking, and we don't want that. So the topoisomerase will alleviate the supercoiling by cutting one strand, passing the other strand through the gap, and then resealing the break. Okay. So here's a short animation of topoisomerase. One thing I have learned in my many years studying biological systems is that when you look at it from the macroscopic level, the design is very apparent. When you look at each of the individual proteins and how they're very specifically crafted to perform their specialized jobs, it takes the design inference to a whole new level. So I just want to give you an example here by doing a deep dive on the topoisomerase enzyme. This is an animation that was produced by Discovery Institute on the topoisomerase.
Wouldn't it be amazing if there were a device that untangled knots automatically? Inside your body, there is. It's called a topoisomerase. It untangles knots in our DNA in an incredible process that keeps us all alive. DNA carries assembly instructions required for your body to generate new cells. But for DNA to do its work, it must be copied in a process known as replication. During replication, the DNA strands are separated, and protein machines use each strand to make a copy of the opposite strand. The end result is 2 copies of the original DNA. However, DNA replication faces a life or death challenge before it can complete its mission. Separating the DNA strands causes twisting in the portion of DNA that has not yet been separated. Eventually, the tension from the twisting increases so much that the uncopied segment of DNA wraps around itself in what are referred to as supercoils. If nothing intervenes, this buildup of supercoils will completely shut down the DNA replication process, since the two strands can no longer be separated, and your cells will die. Enter special proteins called topoisomerases to the rescue. Topoisomerases are amazing molecular machines that untangle knots. They come in two main types. Type 2 typically has three main sections: an upper gate, a middle gate, and a lower gate. Each gate can open or close during the protein's operation. A type 2 topoisomerase untangles DNA in four main steps. First, after two DNA segments enter through the top gate, it uses the middle gate to break one segment of DNA apart. Next, it passes the other DNA segment through this break. Then occurs the crucial step. The topoisomerase recombines the first DNA segment. The result is the elimination of two supercoils. Finally, now that the two DNA segments have been untangled, they are released by the topoisomerase. The second strand is released out the bottom, and the first strand is released out the top. Of course, this is just a simple summary. The actual process is a lot more complicated. Get ready for a more detailed version of how this process may take place based upon the latest research. When we zoom in on the upper part of the topoisomerase, one of the two overlapping DNA segments enters the upper gate. It then binds to the middle gate. Now the second DNA segment enters the upper gate. The DNA in the middle gate is cut in two by the topoisomerase. Two ATP molecules attach to the upper gate and the gate closes. One of the ATP molecules is broken apart into ADP and phosphate, releasing energy that may help keep the upper gate together during the next steps. The middle gate now opens and pulls the two halves of the broken DNA apart, creating a gap. Since the middle gate is connected to the broken ends, the DNA remains attached to it. The second DNA segment now moves through this gap. The middle gate closes and the attached DNA ends are reconnected. The upper gate rotates, which may prevent the second segment from moving back through the break. The remaining ATP molecule breaks apart, and the lower gate opens, allowing the second DNA segment to leave. Then the lower gate closes, the upper gate opens, and the first segment of DNA is finally released. The topoisomerase is now reset, ready to perform the same process all over again. The topoisomerase molecular machine is a wonder of the biological world. Its operation is highly orchestrated, displaying exquisite and intelligent design.
Okay, so that was, yeah.
Yeah, question, how long does that whole process take?
I'm not sure exactly how long, not very long. Knocking at DNA helicase. So the DNA helicase is an enzyme. Who knows what DNA helicase does? Yes.
It splits apart the two sides of the chain.
Yes. So it breaks the hydrogen bonds along the double-stranded DNA molecule, unzipping it and unwinding it, preparing it for replication. Now, if you were to lose the DNA helicase, of course, the DNA cannot separate, and so no copying can take place. Again, we'll zoom in on the DNA helicase and see what it's like, how it does the job that it does.
Helicases separate nucleic acid duplexes into their component strands using energy from ATP hydrolysis. The crystal structure of this DNA helicase from bacteriophage T7 reveals a hexagonal arrangement of 6 identical subunits. Surprisingly, the ring is not 6-fold symmetric, but is slightly squished. A model for the mechanism of how the enzyme might work explains this structural asymmetry. of the 6 potential ATP binding sites, 2 opposing ones bind ATP tightly, 2 are more likely to bind ADP and phosphate, and 2 are empty. These 3 states may interconvert in a coordinate fashion as ATP is hydrolyzed, creating a ripple effect that continuously runs around the ring. Because of these conformational changes, the loops that extend into the central hole of the ring that are proposed to bind DNA oscillate up and down as seen in this cross-section. The oscillating loops might pull a DNA strand through the central hole, thus unwinding the double helix in the process. A frontal view shows the full dynamics of this fascinating protein machine.
And this is, the helicase is spinning the DNA as fast as a jet engine. So it's incredibly fast in unwinding and unzipping the DNA molecule. What about the DNA polymerase? The DNA polymerase is actually performing the copying. See it labeled with the arrows there. So it's doing the copying, and it has its own inbuilt proofreading feature, such that it reduces errors to something like 1 in every 10 to the 8 or 10 to the 9 nucleotide pairs. What about the single-stranded? And remember that the polymerase actually has to be clamped on to the strands of DNA by a sliding clamp, and the clamp has to be loaded onto the DNA also by a clamp loader. So there's a lot of complexity that's not even covered in this slide. There's a single-stranded binding proteins that bind to the single strands of DNA to prevent them from re-annealing during copying. If you knock out the single-stranded binding proteins, the strands are just going to come together during copying. What about primase? Well, the DNA polymerase isn't able to start replicating by itself. It has to be primed, and it's primed by a particular type of RNA polymerase called a primase, which synthesizes a short sequence of RNA from which the DNA polymerase can extend. So without the primase, the DNA replication doesn't get off the ground. As you saw in the animation, because of the anti-parallel nature of DNA with the strands being oriented in 2 directions, in order for the 2 strands to be copied in the same direction, one of these strands has to be copied backwards, and so it actually loops around and is copied discontinuously in short sections. So there are short RNA primers that are synthesized, and then from these primers you get these short sequences of DNA called Okazaki fragments. Eventually the RNA primers have to get removed by exonuclease enzymes, replaced by DNA, and the fragments called Okazaki fragments have to get stitched together, and what glues them together is this class of enzymes called ligases. So if you knock this out, then the Okazaki fragments will remain as fragments. And so as you saw, if you knock out any of these proteins, and there was more, then you don't have a DNA replication machinery that works half as well as it used to or a quarter as well as it used to, but it's broken. So if you want further reading on the argument from molecular machines and irreducible complexity, here's a few books I recommend. Darwin's Black Box by Michael Behe, published in 1996, is the first of his 3 books. Foresight by Marcus Eberlin, who's a Brazilian scientist. Fazali Rana, The Cell's Design, How Chemistry Reveals the Creator's Artistry, and Your Designed Body, which take more of an anatomical and physiological approach, looking at some of the designed features of Human Anatomy and Physiology by Steve Loffman and Howard Glicksman. So, okay. So here are a couple of discussion questions. So how would you evaluate the strength of the argument in this section? Any comments? Yes.
One of the major patterns that you see when looking at biology is diversity.
Mm-hmm.
I mean, the DNA replisome may be one of the exceptions of things where there actually is very low diversity. But don't you also see a lot of different configurations of proteins working together to do a single job task that is done differently in other
Yes.
Other organisms, and does that how does that relate to the fact that each part is necessary, or
Yeah.
yeah.
So you're right. DNA replication is just as an exception, where it's essentially the same whichever organism you're looking at. And what's fascinating about DNA replication is that there's academic research that has shown, happy to send out papers on this if anyone's interested, that the proteins involved in the replication of DNA in the lineage of archaea and eukarya and the lineage of bacteria are not homologous, even though they perform the same functions. And so this is an extreme case of convergent evolution, which I think is actually quite powerful evidence of design. Now, you're completely right that for many systems, you find a diversity of ways of performing the same task. Bacterial flagella, which is a classic example of intelligent design, one of the flagship examples that people often cite, is such an example, right? There's a lot of different flagella out there, each with their own set of parts and so forth. I would argue that, so I don't think about irreducible complexity in terms of the identity of the structural components per se, but rather in terms of the functions that need to be performed, right? So you can perform, in principle, the same function using two different proteins versus one protein in another system, right? Or you might perform, you might have one protein that performs multiple functions, that has different domains that perform different functions and so forth. So I think that's a more helpful way of thinking about irreducible complexity than simply thinking about the structural components. And I think that when you do that, you see that when you have different ways of achieving the same goal, all you're seeing is just different ways of achieving the same function. But the core minimum set of functions still needs to be performed, right? Does that make sense? Any other questions? Yes?
Another question I'm thinking about. So, like, if we were walking along and we found a watch on the ground,
Yeah.
we would know that that watch is highly complex. It
Yeah.
had to be made by a complex being that had foresight and was able to think about all those components working together. In the same way that humans are a complex organism that was made by a higher complex entity that was thinking about all that and foresight and so much planning and everything going into that. So then I guess this question might kind of go beyond the scope of this talk, but what made God? Or like, you know, if God is that entirely complex, then how did God come to be?
So a few points here. First of all, in order to postulate an explanation as the best explanation for a given phenomena, that doesn't require you, logically, to have an explanation of the explanation, right? So for example, if I want to explain a hieroglyphic inscription, then the best explanation is intelligence, even if I have no idea where the particular civilization came from. I have no idea of their genesis story or their origin story, right? Or if I want to explain the existence of a pocket watch, I have no idea like who the designer is, who their parents were, where they came from, but I nonetheless have a good justification for design, right? So that's a logically downstream question, which is an interesting question, sure, but you don't have to be able to answer it before you can answer this question. Secondly, if we're talking about theism, which I think a theistic designer is by far the best candidate for the role of the designer, and we're postulating that God is eternal, right? God doesn't fit into the category of things that are made, things that are finite, right? So the Kalam cosmological argument, which is a famous argument for God, doesn't say that everything has a cause, but rather everything which begins to exist has a cause. So there are two sorts of things. There are contingent things, like ourselves, so we don't have to be here, right? We have a beginning. And then there's non-contingent things, and God would be a non-contingent thing. And classically, God is understood as being very simple. He's not comprised of material parts. So it's not at all obvious to me that God requires an explanation in virtue of his complexity, because God isn't even made of parts. So it's very difficult to talk about God as being complex materially, right? Yeah.
So this is more of a question about DNA polymerase specifically, because when I think of making arguments from irreducible complexity, one argument I've made in the past, I'm thinking of ways where if I take out this piece at the point where that trait mutated or evolved, whatever term you want to use, if it's a multi-part system that works together and one part is missing because it hasn't evolved yet, the organism just dies. Does that kind of thing work with DNA polymerase in the same way?
It does. So this brings me to the second discussion question, which is, can you think of any additional reasons why self-replicatability is particularly difficult to account for by evolutionary processes? So beyond what we've discussed here, can anyone think of why this would be? This is an example of what I call irreducible complexity on steroids, and it relates to that comment. Can anyone think of a reason? Well, self-replicatability is essential for differential survival. And differential survival is in turn a prerequisite for natural selection, right? So you can't invoke natural selection to explain the origins of self-replicatability without assuming the existence of everything you're trying to explain, right? There are other examples like this, like the bacterial cell division machinery, for example, I also think is irreducibly complex. So it's very difficult to appeal to natural selection to explain a process like this, even aside from the irreducibly complex features. Okay, any comments or pushback before I move on? Okay. So let's move on to our final section then, which is on the waiting time problem. So I want to begin by this is essentially the mathematical expression of irreducible complexity. So I want to define, first of all, coordinated mutations. What do I mean by coordinated mutations? Well, coordinated mutations are where you have two or more coincident genetic changes that only together produce an adaptive phenotypic effect that allows for selection to operate, okay? So when you require two or more coincident changes in order to get an adaptive benefit, that is what I mean by coordinated mutations. So what is the waiting time problem? Well, all mutations have two time constraints that depend on population size and generation time. These are, number one, the waiting time for a mutation to occur by chance, randomly in the population. And secondly, the waiting time for the fixation of this mutation. That is to say, it goes from being the lone individual out there that carries that mutation to becoming the norm that gets established in the population, right? That's what we mean by fixation. Now the question that we can ask is, does the history of life provide the resources for evolution to accommodate these waiting times? So what is the waiting time dilemma? Well, if you have a large population size, the waiting time for a mutation to occur decreases because there's more individuals for it to happen in randomly by chance, right? But the fixation time increases because there's more individuals for the mutation to sweep through, right? With small population sizes, on the other hand, the waiting time for a mutation to occur increases because there's fewer individuals for it to randomly happen in by chance, so it's going to take longer. But fixation time decreases because there's fewer individuals for the mutation to sweep through. Thus, there is no easy way for evolution to work around the waiting time problem, whether you have a large or a small population size. Now, I'm just going to give you one case study. There's a lot more I can say on this topic, but I'm just going to give you one case study. This is from The Edge of Evolution by Michael Behe. So malaria is a disease that affects many countries in Africa. It's borne by mosquitoes. It's transmitted by mosquitoes, in particular pregnant mosquitoes, and is caused by a particular parasite. Does anyone know the Latin name for the parasite? No, Plasmodium falciparum, right? And there are two main drugs. There are two major drugs that are used in treating malaria: chloroquine and atovaquone, right? And to acquire resistance to atovaquone, malaria only requires one mutation, right? And so it arises roughly once in every third sick persons, given how many malarial parasites there are in the person's body. But for chloroquine, malaria requires two codependent amino acid substitutions in order to acquire resistance to chloroquine, right? Public health data suggests that it occurs, that is, malarial resistance to chloroquine occurs approximately once in every 10 to the 20 cells, malarial cells. And so, it arises in approximately every billion sick persons, right, given how many parasites there are in sick persons. And given that there's about 1 billion people with malaria in any given year, you have this arise. You've got approximately one person at a time in every year that has malarial resistant to chloroquine in their body. From this, it may be predicted that an adaptation requiring 4 codependent substitutions would arise in every 10 to the 40 malarial cells. So that's 10 to the 20 squared, so 10 to the 40. So given that less than 10 to the 40 organisms have likely ever existed on Earth, this number is quite prohibitive. So Mike Behe, in his book The Edge of Evolution, notes that the ratio of humanoid creatures in the past 10 million years to the number of parasites needed for chloroquine resistance is 1 to 100 million. And for most of the past 10 million years, the population of the line of primates leading to humans is thought to, at best, to have been roughly about 1 million or so. It's only in the past few thousand years that the number has accelerated up to today's population size. And so he notes in his book The Edge of Evolution, I quote, "On average, for humans to achieve a mutation like this by chance, we would need to wait 100 million times 10 million years, that's 10 to the 15 years. Since that is many times the age of the universe, it is reasonable to conclude the following. No mutation that is of the same complexity as chloroquine resistance in malaria arose by Darwinian evolution in the line leading to humans in the past 10 million years." So if you want more on this, I recommend these books, The Edge of Evolution and Darwin Devolves by Michael Behe, and Douglas Axe's book, Undeniable. Okay, discussion questions. How would you evaluate the strength of this section? Yes?
Are these times consistent across different organisms?
So we're talking particularly in this case about malaria, and so the waiting times are going to be based on the mutation rates, the population sizes, and the generation turnover times, right? These, of course, are different in different organisms. In humans, we have lower mutation rates, much longer generation turnover times, and much smaller population sizes than malaria. So it's going to take us longer than the malarial parasite to acquire the same complexity of adaptation, right? Because we have fewer individuals and we have much longer generation turnover times. Yes?
This argument assumes that we're looking for two or more, but two specific mutations that get to a desired function.
Correct.
So wouldn't the probability of some change and even some positive change be much higher over the same time period? Because it's not necessarily looking for a specific change.
Right. Yeah, so there are many adaptations that are within the limitations of the evolutionary process. So there are many changes that require one coincident mutation. We saw in malaria, it does acquire resistance to chloroquine, right? And because you have the large enough population sizes, and as the public health data indicates that it arises roughly once every 10 to the 20 cells. And so it does. I mean, to take another example. There's a particular mutation. It's called I think it's the HSB mutation, so have you heard of the heterozygous advantage, where you have if you're heterozygous for malaria, then if you're heterozygous for the sickle cell anemia, then you have resistance to malaria, which is known as the heterozygous advantage. And also, if you have the sickle cell anemia, you're resistant to malaria, even though it has significant drawbacks with having sickle cell anemia. There's another mutation which confers the same benefits to the sickle cell gene, but doesn't have the same drawbacks as malaria, right? And it requires the mutation of sickle cell anemia plus another one, right? It rises so rarely in humans. As far as I understand, it hasn't arisen in Africa. There's one case that was documented in New York where it arose. But the times that it arises because the first mutation confers, the first mutation is the sickle cell mutation. The second mutation, gives you this additional advantage, right? But you don't get them you only have the population of humans that have sickle cell to work with, because it requires that mutation which has an advantage and then the second mutation. It doesn't arise where you get the two mutations happening together bypassing entirely the sickle cell advantage. So it's very difficult to get these genetic combinations where you require multiple codependent mutations to happen simultaneously in a coordinated fashion to acquire some complex advantage. And it's even more difficult if you need like 4 coincident genetic mutations. Then the probability is just multiplying exponentially. Does that make sense?
Yeah, so this is specific to irreducible complexity,
Right.
at least irreducibly complex like systems,
Yeah.
not just evolution
Yeah.
in general.
Yeah. That's the B Harlan mutation is one I'm thinking. Yeah, go ahead.
So that example that you just gave was like was the case where in order to get for the second mutation to have a lot higher fitness value, you need the first one prior, and that one first one is unlikely. The second one is very unlikely. But
Yeah.
could it not be the case where there are chains of likely mutations that eventually lower the probability of the last one that is unlikely in the very first genetic environment, so there's like a long chain that eventually lowers the probability to where it can happen. But all of those mutations along the way are likely to happen, but there's just enough of them that you wouldn't be able to predict that it would reach that point beforehand.
Yeah, I mean, the vast majority of interesting changes require multiple codependent mutations to arise. And so it's difficult for me to conceive of that being a way of explaining most cases of complex genetic mutations. And furthermore, most mutations, so about 3/4 of all mutations are neutral selectively. They're neither here nor there in terms of the organism's fitness. Of the remaining quarter of mutations, most of them are selectively neutral. Most of them are harmful. And there's only a very small minority of mutations that are beneficial to the organism. All of that very small, so antibiotic resistance, insecticide resistance, and so forth would be in that minority of mutations. Of those beneficial mutations, the vast majority of them are harmful or destructive mutations rather than gain-of-function mutations. So that's also a significant problem that affects this as well. So, most cases of antibiotic resistance involve, say, modifying a target site for an antibiotic or something like that, so it can no longer bind, etc. So, if you want to conserve battery on your cell phone, you can turn off the GPS, right? But that's turning something off rather than turning something else on. Makes sense. Okay. Can you think of any analogies that would help to explain the argument of this section? Yeah.
I guess I still just kind of had a general question. So, for your background as an evolutionary biologist, do you kind of see evolution as something that's happening but needed kind of God's kickstart to help with some of these key mutations early on to allow humans to exist at this point in the time scale? Or do you see God as being like a being that placed Adam and Eve literally?
Yeah, I think Adam and Eve were specially created.
Okay, gotcha.
Yeah. Okay. So, anyone think of an analogy? Yeah.
Simple probability. I mean, if you flip a coin, getting ahead once is not all that unlikely, but getting ahead 10 times in a row is a lot more unlikely, and so on and so forth.
Yeah, exactly. Or an analogy I like sometimes to give is imagine an ATM machine where there's 10 digits, 0 to 9, and there's 4, and there's if you've stolen someone's bank card, but you don't know the PIN number, right? How many ways are there of dialing in a 4-digit number on a 10-digit PIN pad? Anyone know? 10 to the 4th power, right? 10,000 possible ways. And so if you only have 3 trials before the bank locks you out of your account, that corresponds to what we call probabilistic resources, the number of attempts you have to get it right. Whereas if you only have 3 attempts, you're overwhelmingly more likely to fail when you only have a 1 in 10,000 chance of getting the right PIN number. So that's I think a good analogy here. Okay, so in conclusion, a life-permitting universe is unlikely on naturalism. Irreducible complex systems challenge naturalistic accounts of life's origins. The waiting time problem offers a mathematical expression of irreducible complexity. Intelligent design predicts these observations better than does naturalism. Okay, I'm going to finish with a couple of texts from Scripture. This is from the Apostle Paul in the book of Romans, chapter 1, verse 20. He says, "For God's invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world, and the things that have been made, so they are without excuse." Psalm 19:1 says, "The heavens declare the glory of God, and the sky above proclaims his handiwork." And how vindicated the Apostle Paul and the psalmist would feel were they alive today in view of our modern advances in physical and life sciences. I'll remind you of my contact details if you want to reach out privately at jmclatchie.discovery.org. You can also find my personal website up there for more essays, articles, videos relating to intelligent design, as well as biblical scholarship, theology, apologetics. And again, I remind you about the service talkaboutdoubts.com if you would like to reach out for private mentoring with regards to doubts about the veracity of Christianity. Thanks for your attention. Happy to take any additional questions.
Okay, I just want to thank everyone for coming.